Hello everyone, welcome back to my channel. I'm here to take you through another biology lesson. So it's your favorite biology teacher, Najuma Sharok. Earlier we learned how to use microscopes. We looked at the light microscope and an electron microscope. And in today's lesson, we are going to concentrate on the ultrastructure of a cell, ultrastructure of an animal cell, and ultrastructure of a plant cell. When someone talks about ultrastructure, what does that really mean? We are looking at the fine details, the details of what a cell consists of, the different features that are going to be found within a cell, especially as seen using an electron microscope. Remember that a light microscope had a limit on how much details we could see. When it comes to an electron microscope, it reveals so much more detail. Actually, when scientists first observed a cell using an electron microscope, they were amazed by how much detail they could see. And that takes us to our first question. We should be able to identify structures that can be seen with an electron microscope, but you can never see them with a light microscope. So let's compare an animal cell as seen under a light microscope and an animal cell as seen under an electron microscope and see the difference. So here on the left, we have an animal cell as seen under a very high quality light microscope. On the right, we have an animal cell as seen under an electron microscope, particularly a transmission electron microscope, because we can see inner features. Let's start with, we can start with any feature. Let's start with, which feature should we start with? Let's start with the nucleus. The nucleus. Now, when you look at this nucleus, you can see that in the light microscope shows us that the nucleus is made up of, yes, a membrane, just a single membrane, surrounded by a single membrane, which we call a nuclear membrane. When you get back here to the electron micrograph, we can see that, yes, there is a nuclear membrane, but that nuclear membrane is double. You can see we have this first line here and then this other line. So it's a double membrane. This side, you'll never see it. Here, the details are shown. It's a double membrane, and such a double membrane is called an envelope. Two membranes surrounding this organelle are nucleus. So a nucleus has an envelope. Envelope means double membrane. Next, we can see their nuclear pores. The nucleus has pores. However, under a light microscope, you never see those pores. We can see that the nucleus has an extension. The nuclear membrane extends outwards to form the rough endoplasmic reticulum. You can see this extension of the membrane, yes, into the cytoplasm. This side, you can see it's non-existent. If you're using a light microscope, you'll never be able to see it. We can see these tiny black dots, the ribosomes. The ribosomes on the rough endoplasmic reticulum and also in the cytoplasm. Go to a light microscope, you will not see the ribosomes. We see lysosomes. Light microscope, you never see lysosomes. Microvilli are visible in an animal cell. This side, you never see microvilli if you're using a light microscope. Look at the mitochondrion. We are shown the mitochondrion has an outer membrane and it also has an inner membrane which is folded. That is as seen under an electron microscope. I think you can see it here, the mitochondrion. Whereas if you go and look at this light micrograph, the mitochondrion, yes, you can see it, but it's like it has one membrane just enclosing it. So this side, the mitochondrion has an envelope, which simply means two membranes. We can see that we have centrioles, and there are two centrioles. This side, we can still see a centriole, but it shows us as if it's one centriole. We can see the Golgi vesicles. Even this side, you could see them, Golgi vesicles. You could see the smooth endoplasmic reticulum under an electron microscope. This side, you never see the smooth endoplasmic reticulum. So you should be able to tell those differences. Structures that you can see when using an electron microscope and structures that you can never see when using a light microscope, or structures that you can see in both. Yes, and sometimes in papers, you'll be expected to make drawings. So look at these diagrams that are given to you. Be very familiar with the different structures that are found in these micrographs that you're given, and make sure that you can be able to make drawings and even make annotations. Like you could be told to draw the rough endoplasmic reticulum. You should know how am I supposed to be able to draw it. So make sure that you revise the diagrams and you're well conversant with the different structures that are found within a cell. So right now we are going to look at the uses of each and every structure that is found within a cell. Without further ado, let's start with the cell surface membrane. I'm sure everyone knows what the cell surface membrane is. The cell surface membrane is that outer layer enclosing a cell. It holds all the cell contents together. This structure here, the cell surface membrane, holds all the cell contents together, and its major role is to control exchange between the cell and its environment. The cell surface membrane is also partially permeable. Partially permeable simply means that it allows some materials to pass through and enter into a cell, and it prevents other materials from passing or moving out of a cell. If this is our cell, materials to move in have to pass through the cell surface membrane. If they are to move out, still they have to pass through the cell surface membrane. This is our cell surface membrane. But the cell membrane does not allow everything to enter. And at the same time, it does not allow everything to leave. So its role is to control exchange between the cell and its environment. It determines what enters the cell or leaves the cell. This cell surface membrane is extremely thin. Its size is around 7 nanometers. We looked at these units of measurement. Hope right now you are very familiar with them. If you're not familiar with them, you can go and check our first video that we made. It was about units of measurement as used in microscopy. Now, when it comes to these sizes, don't be surprised if you go into a paper and they ask you what is the size of the approximate size of the cell surface membrane. So please take note of that. The cell surface membrane is 7 nanometers. They can really ask for details like that. When you look at this cell surface membrane under very high magnifications, it appears to have a trilaminar appearance, which means it appears to be made up of 3 layers, like you can see here. This is outside the cell, and then maybe this is inside the cell where we have the cytoplasm, and the outside is separated from the cytoplasm by the membrane. This is the membrane here. You can see we have this layer, a middle one, and this other layer. So it appears to be made up of 3 layers: outer, middle, and the inner layer. Later, as you learn about cell membrane and transport, you understand why the cell surface membrane appears to be made up of 3 layers, why it has the trilaminar appearance. We are done with the cell surface membrane. So let's go to the next organelle, which is microvilli, or microvillus if it's one. When there are many, we call them microvilli. So microvilli are finger-like extensions of the cell surface membrane. Not all cells have microvilli. No. Microvilli are going to be found in just some cells, especially cells that line structures where absorption occurs. Like an example, cells in the ileum have microvilli. Cells in the kidney, where we have reabsorption of water, they have microvilli. So these microvilli simply increase the surface area for absorption. surface area for absorption. Because if materials are to be absorbed into this cell, maybe by diffusion or active transport, these microvilli are going to provide a large surface area because all over these villi, it means that materials are going to be absorbed. And that simply means that the process of absorption is going to be faster. It could be diffusion, it could be osmosis, it could be active transport. So that is what the microvilli do. Microvilli are extensions of the cell surface membrane, and they are mainly found in animal cells. Okay, let's look at the next organelle, which is the nucleus. I'm sure all of us know what a nucleus is, but if you don't know what it is, still, I'm going to explain to you what the nucleus is and what its role is. Now, this is, most people refer to it as the brain of the cell, or some people call it, most people basically brain of the cell. And what does it do? But I don't want you to write in a paper that the nucleus is the brain of a cell. Obviously, no one will ever give you a mark if you write that. But just know that it's an organelle that controls cell activities. Now, we have seen the nucleus is surrounded by two membranes, which you call an envelope. You can see this inner membrane and then the outer membrane. However, sometimes the outer membrane extends outwards, like you can see here, to form the endoplasmic reticulum. Inner membrane, outer membrane extends outwards to form the endoplasmic reticulum. Yeah. Now, inside this nucleus, the nucleus has DNA, so it encloses DNA or genetic material. This means that this is where you're going to find the chromosomes, because chromosomes contain DNA. And when you look into this nucleus further, you're going to find that it has what we call a nucleolus. We shall look at the use of the nucleolus later. And most of the time, the nucleolus is much more stained compared to the contents, to the other contents of the nucleus. The nucleus has pores. Now remember that whichever organelle has a membrane surrounding it, the membrane performs the same role. That membrane still controls exchange between that organelle and the surrounding. Like now, the nuclear membrane will be controlling exchange between the nucleus and the cytoplasm. Now, the pores, the nuclear pores allow materials to enter into the nucleus or move out of the nucleus. We should know examples of molecules that can enter into a nucleus and leave the nucleus. Like an example, you could find that we can have nucleotides. You'll learn about nucleotides later and nucleic acids and protein synthesis. But nucleotides can enter into the nucleus. ATP, which is an energy carrying molecule, can enter into a nucleus. Now we can have other molecules that are moving out of the nucleus, maybe such as mRNA. You will learn about mRNA. If not, you've heard about it already. But for now, just know it's an example of a molecule that can get out of a nucleus. We can have ribosomes getting out of the nucleus. We are going to learn about ribosomes still. So those are some of the structures or molecules that can enter or leave the nucleus. So nuclear pores allow exchange between the nucleus and the cytoplasm. Now, the nucleus is said to control cell activities, but how does it control cell activities? It does so simply by housing DNA. So the DNA contains genes, and those genes are responsible for controlling all the activities of a cell, and they determine and control inheritance. So because the nucleus has DNA, which controls all the cell activities, that is why it is said that the nucleus controls cell activity simply by having DNA. So let's look further. This is still a nucleus. Now, this is a transmission electron micrograph. You find a lot of diagrams or pictures that are in black and white. Even in your papers, most of the pictures are in black and white because they're coming from electron microscopes. And you should be able to identify organelles. They will just label any organelle, ask you about its role, or ask you questions about it. Like in this class, sorry, in this case, we are looking at the nucleus. So this is our nucleus. This is the nucleolus. You can see the outer membrane and the inner membrane here. You can see the pores, nuclear pores. You can see the nuclear pores. So in here, this is where you're going to find the chromosomes or chromatin and the genes which are found on DNA. Now, we can see the endoplasmic reticulum here. This is these membranes, these flattened membranes form the endoplasmic reticulum. You can see it outside the nucleus. So in a nucleus contains DNA, and DNA can be organized into chromosomes. So DNA can exist in two forms. It can exist as chromatin. In this picture, when you look at chromatin, chromatin is loosely coiled DNA. You can see here it's thread-like and not so much organized. So this is chromatin. However, when a cell is about to divide, that chromatin is condensed into what we call a chromosome. Because remember, during cell division, the DNA has to be divided into the daughter cells equally. So if it's in the form of chromatin, it cannot easily be divided equally. So that's why it has to be condensed and coiled into chromosomes such that division of this DNA amongst the daughter cells is very easy, whereby one chromatid will go into one cell, another chromatid also goes into another cell. You learn about the details of that instead in another topic. Yes, but from here, just know that DNA can be in form of chromatin or in form of chromosomes. And when you look at this DNA in animal cells, that DNA most of the time is wound around proteins called histone proteins. Still, you learn about these details under nucleic acids and protein synthesis. But for now, just know that DNA is found within a nucleus, and DNA controls activities of a cell because it has genes which determine which proteins are going to be made in your body, and those genes still determine inheritance. Within a nucleus, we've seen there is a nucleolus. This structure here is called a nucleolus. Usually it stains more compared to the other parts of a nucleus. So what is the use of a nucleolus? Its use is to simply make ribosomes. Yes, the nucleolus makes ribosomes. We are going to look at ribosomes. And within a nucleus, you could find that sometimes it can have just one a nucleus could have one nucleolus, or it could have more. Yes, it can have one or more nucleoli. And when you look at that nucleolus, it contains DNA, which DNA has genes that it uses to form ribosomal RNA. Ribosomal RNA makes up part of the ribosomes. So when the nucleolus is making ribosomes, it combines ribosomal RNA and certain proteins to form a ribosome. So if a cell needs more ribosomes, that means that the nucleolus in the nucleus will be bigger in size so that it can make as more ribosomes as possible. And you find that during formation of ribosomes, the nucleolus will only be present in a nucleus if there is need to form ribosomes. If there is no need to form ribosomes, usually the nucleolus is not present. Like during cell division, mitosis, you learn that the nucleolus sometimes disintegrates. it will disintegrate when there is no need to form ribosomes. If there is need to form ribosomes, then the different parts of it come together so that ribosomes are made. So we are done with the nucleus. Let's look at the other organelle, the endoplasmic reticulum. The endoplasmic reticulum. So the endoplasmic reticulum is an extensive system of membranes, just membranes, but those membranes appear like flattened sacs. A flattened sac, you can relate a flattened sac to a new polythene bag that has not yet been opened. Most of the time it's very flat, and you might not know that there is a cavity within it. But when you open it up, you'll find that there is a cavity where you can even put some things of yours that you would want to put there. So even the endoplasmic reticulum is made up of flattened sacs or flattened compartments. And maybe just going back to this diagram here. This is the endoplasmic reticulum. We have seen that it's an extension of the nuclear membrane. But sometimes it can still exist within a cell and it's not extending from the nuclear membrane. Yes. So when you look at those flattened sacs, within them, processes can take place inside there without interruption from the contents of the cytoplasm. So there are two types of endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. In this case, this electron micrograph shows us the rough. Why is it called the rough? It is called the rough because it has those tiny black dots on it. I don't know whether you can see them, the tiny black dots. Now, those tiny black dots on the outside are called ribosomes. So it appears, you know how you can have your face and maybe it has some pimples on it? Someone can say that your face is rough. Why? Because it has pimples. So even when it comes to the RER, because of those ribosomes, that is why it is called rough. Rough endoplasmic reticulum. Yeah. So this 3D diagram shows you those flattened sacs I was telling you about. So this is the rough endoplasmic reticulum. These are the ribosomes attached on its outside. So still, this is the rough endoplasmic reticulum. You can see those black outside dots are the ribosomes. So what is the use of the rough endoplasmic reticulum? Now, because it has ribosomes covering it, and ribosomes carry out protein synthesis, we can still say that the rough endoplasmic reticulum carries out protein synthesis, or it manufactures proteins. And most of the time, once the proteins on the ribosomes have been made, those proteins can enter into these sacs and start moving through those flattened sacs. As the proteins move through the flattened sacs, the proteins get modified. They get modified. Changes happen to those proteins. Some of the changes could be that maybe some carbohydrates are added to the proteins to form glycoproteins. And later, the proteins will leave those sacs. So for the proteins to leave the sacs, usually they live in form of vesicles. So a vesicle develops on the outside of this RER, and then it buds off. So within that vesicle, we shall have proteins being carried from one part of a cell to another. So that is why if you asked for the uses of the rough endoplasmic reticulum, number one, mention that it carries out protein synthesis simply because of the ribosomes it has. Secondly, it modifies proteins, it processes proteins, it packages proteins. Yes, it packages proteins and processes proteins. And those vesicles that bud off from this RER are used in secretion. Remember, secretion is releasing substances either into the cell or out of the cell. Like this vesicle could go fuse with a cell surface membrane and pour the contents outwards by exocytosis. Some of these things I'm talking about might be new, but as we go on, you'll be understanding them. Because remember, this is our first topic, so some of the information I'm talking about is in other topics that you're yet to learn about. But just know that the vesicles form part of the secretory pathway. Why? Because they're going to be carrying those proteins from the rough endoplasmic reticulum to other parts of a cell where the proteins are needed. The other type of ER is the smooth. The smooth is here. You can see the smooth endoplasmic reticulum has no ribosomes. Yeah, it's very smooth. It has no ribosomes. So what is its use? It makes lipids. It makes steroids. I'm sure you've heard about reproductive hormones or estrogen, testosterone, and those are found in the smooth. They are made in the smooth ER. The smooth ER also stores calcium ions, and that is particularly the smooth ER in your muscles. It stores calcium ions. Then in the liver, the smooth ER is involved in drug metabolism. So those are the major uses of the smooth endoplasmic reticulum. Sometimes it could be hard for you to get all the uses, but at least get two of them. Get two of them. And yes, you'll be good to go. Next, we have the ribosomes. We have seen the ribosomes already earlier. We have seen that ribosomes can be found in the cytoplasm. An example you can see here, some of the ribosomes, those black dots, are just free in the cytoplasm. And then others, you can see they are attached to the rough endoplasmic reticulum. So they can either be found free in the cytoplasm or they can be found attached to the rough endoplasmic reticulum. So what is the use? Ribosomes are some of the smallest organelles, and they are roughly 25 nanometers in diameter. Now, at very high magnifications, you'll be able to see that ribosomes are made up of a large subunit and a small subunit. They appear like this. You have a large subunit, sorry, a small and a large subunit. So here they are trying to show us the structure of a human ribosomes. This is one subunit, and then this is another subunit. So these ribosomes have different sizes, and the size of these ribosomes is measured in S units. I'm not so sure how to pronounce this word. But yeah, those are the units that are used to measure the size of very tiny cell structures like the ribosomes. No one will ask you about the details of why those units are used, but just know that those are the units that are used to measure ribosomes. So some ribosomes are large, like the ribosomes in animal cells are large, so they are called ATS ribosomes. ATS ribosomes, and then those in prokaryotes in the bacteria are small, so they are called 70S ribosomes. Ribosomes are made up of ribosomal RNA and protein, and we even saw that these ribosomes are made by the nucleolus. Yes, the nucleolus makes those ribosomes. So these ribosomes are the regions where protein synthesis occurs. They manufacture proteins. How do they manufacture proteins? By joining amino acids together. You'll still learn about the process of protein synthesis, how proteins are made. But from this section, just know that ribosomes are used in making proteins. So ATS ribosomes make proteins in animal cells, in plant cells, in fungal cells, in eukaryotes, whereas 70S ribosomes make proteins in prokaryotes, which are the bacteria. Hope we are still on the same page. Hope we are still on the same page. Yes, I do offer one-on-one sessions if anyone is interested. Yes, I do offer one-on-one sessions. I will leave my email address in case you want to contact me for such, so that we can get in touch and ensure that we get that ester in biology. Now, this diagram shows you how proteins are moved from one part of a cell to another. So here we have a ribosome, or a ribosome that is found on the RER, assuming this is our RER, where the ribosome manufacture proteins. We mentioned that the proteins eventually enter into the RER where they are modified, and later they have to leave. As the proteins leave, I told you that they live in form of vesicles. So an outgrowth grows on the RER and it buds off and breaks off and forms a vesicle. What is a vesicle? A vesicle is like a small sac or a small vacuole. Yeah, so a small sac that is enclosing some different materials, like in this case, it's enclosing the proteins. And once these vesicles leave the RER, most of the time they go to another cell organelle known as a Golgi body or a Golgi apparatus. So the vesicle leaves the RER and it fuses with the Golgi apparatus. You can see in this case, this is the Golgi apparatus. A Golgi apparatus is also a system of flattened sacs, but the flattened sacs appear to be stuck one on top of the other. This diagram shows us the Golgi apparatus. You can see it. The Golgi body appears to be made up of a stack. It's a stack, one flattened sac on top of the other. And when you look at the Golgi body, it appears to be made on one end, and on another end, it's being broken down. What does that mean? Now, here they are showing you an incoming vesicle. So the vesicle will come and join and pour or release its contents into these sacs. So the proteins will start moving through as they get further modification. Then if the proteins are to leave, they will leave through the vesicles still. So an outgrowth forms and then the vesicle buds off. So that takes us to the Golgi body. This is still from a microscope. It shows us the Golgi body. As I told you, it's a stack of flattened sacs. It appears to be made on one end as the vesicles join, and then on another end as the vesicles leave, it appears to be broken down. So what are the uses of the Golgi body? Number one, it collects those proteins from the vesicles which came from the RER, collects those proteins, processes them, sorts them so that they are ready for transport. And how are they transported away from the Golgi body? They are also transported in form of vesicles. So you hear someone talking about maybe Golgi vesicles. Those are simply vesicles that are coming off the Golgi body. Those vesicles can be used in secretion. So how are these proteins, particularly, how are they modified? Number one, sugars can be added to those proteins. Remember, proteins are only made up of amino acids, but there are certain proteins that have carbohydrates attached to them. So what will happen is that addition of those carbohydrates to the proteins to form glycoproteins will happen in the Golgi body. Usually proteins or every polypeptide has the first amino acid being methionine. So within this Golgi body, that first amino acid is always removed so that the protein can carry out its function. And that is done in the Golgi body. So it modifies proteins. It packages proteins because it will package them in vesicles. It processes them. But it has other uses. Let's go into the other uses. The Golgi vesicles formed, some of them make what we call lysosomes. Oh, sorry about that. My sharing is paused. Okay, hope we are back and you can see my screen. Yeah, Golgi vesicles form lysosomes. We are going to look at what lysosomes do. So the Golgi body does not only modify proteins, it also modifies lipids. Those are the fats and oils. How? Still by adding carbohydrates to them to form glycolipids. Golgi enzymes in plants are involved in synthesis of new cell walls in a plant. Remember how cell walls are very important in a plant cell. So Golgi enzymes are involved in synthesis of new cell walls in plant cells. Within our gas exchange system, the goblet cells release a substance called mucin. Mucin is used in making mucus. You know how our gas exchange system needs the mucus to trap pathogens or dust that comes along with the air. So that mucin, which is used to make mucus, is made from the Golgi apparatus in those goblet cells. So those goblet cells have a lot of Golgi bodies so that they can produce as much mucin as possible and make as much mucus as possible. Next organelle, we have the lysosomes. So lysosomes are also small sacs. Remember, we have seen lysosomes come from the Golgi body. So some of these sacs that bud off from the Golgi body, some of these vesicles form what we call lysosomes. So lysosomes are very special for us. Why? They are used to hold digestive enzymes. So they contain digestive enzymes. A cell might need to destroy some structures within it. So what will it use? It will use those digestive enzymes or hydrolytic enzymes. And most of the time, these enzymes work best under an acidic pH. So this means that within a lysosome, those enzymes are provided with the particular pH they need, which is a pH of closely 4 to 5, whereas the cytoplasm has a pH of around 6 to 7. So this lysosome will contain enzymes which break down enzymes that are digestive, such as such as proteases, lipases, nucleases, where proteases will be used in breaking down proteins. Lipases will be used in breaking down lipids. Nucleases will be used in breaking down nucleic acids. And this diagram shows us a special lysosome, which is found in a sperm. Yes, you learned about reproduction, and you know that a sperm is a male gamete. For the sperm to enter into an egg, it has to first digest the membranes, the layers covering an egg. So this acrosome has those digestive enzymes which digest the egg membrane before the sperm enters to release the nucleus. So those lysosomes, so anacrosome is a special lysosome found in sperms. Other uses of the lysosomes are used in getting rid of unwanted components. An example, you could have organelles that are no longer functioning. It could be maybe an old mitochondrion or an old chloroplast. So the lysosomes can engulf it and destroy it with the digestive enzymes that they contain. Lysosomes are used in endocytosis. You learn about details of endocytosis still in chapter 4, cell membrane and transport. But this picture shows you what endocytosis is about. When pathogens enter into your body, like the bacteria, we have white blood cells, particularly the phagocytes. So the phagocytes enclose the bacteria and place it into a vacuole known as a phagocytic vacuole. And once the bacteria has been enclosed in a vacuole, lysosomes come fuse with that vacuole and release digestive enzymes, which break down the bacterium. So lysosomes are used in endocytosis, for example. by the white blood cells as they destroy pathogens. Next, I've talked about the acrosome, which is involved in exocytosis. Endo, it means they are used inside a cell, endocytosis. With exocytosis, the digestive enzymes are released outside. Like an example, if this is our egg cell that is to be penetrated, this acrosome will release digestive enzymes onto. So the digestive enzymes are being released outside the cell. So that will be exocytosis. They'll be released outside the cell to digest this egg membrane. So that is exocytosis. Then lysosomes can also be used in self-digestion. Under self-digestion, the whole cell can kill itself, if I may bring it in simple terms. It can just release all the digestive enzymes into itself and then it is destroyed. A whole cell can be digested. So in this case, the contents of the lysosomes are simply released into the cytoplasm, and then the whole cell is digested. That is what they call autolysis. So autolysis happens in different conditions, like sometimes a cell could divide to form daughter cells, and maybe those daughter cells might turn into cancerous cells. So usually, those daughter cells that might turn into cancerous cells can just be killed through autolysis so that you do not develop cancer. So that means that if cancer develops in an individual, those cells were able to bypass the system that brings about autolysis. That's why they didn't get killed. Okay, so we can continue with other organelles. Hope you guys are still following. Hope you're still following. Feel free to take some water. Let me also take some water. So this is an example of a question. We are having a figure showing a diagram based on an electron micrograph of a secretory cell from the pancreas. So they're telling us this type of cell is specialized for secreting proteins, meaning releasing proteins outside the cell. Some of the proteins are digestive enzymes of the pancreas, which are found in pancreatic juice. So the cell is very active, requiring a lot of energy. The arrows show us the route taken by the protein molecules. Like in this case, they are showing you the arrows taken. The arrows show you the route taken by the protein molecules. Okay. So they are telling us, describe briefly what is happening at each of the stages. So we have stage A, we have stage B, we have stage C, we have stage D. Stage A is here, and it has even been magnified so that you can really see what is happening at stage A. They are showing us we have a ribosome. Remember, this is the RER. And an arrow is showing us that something is coming from the ribosome entering into this flattened sac, the RER. Then we have B. B is showing us we are having a vesicle moving to join this structure, the Golgi body. So you need to be able to identify the different organelles. Like in this case, I expect you to be able to identify the Golgi body, which is here, to be able to identify the rough ER, which is here, to be able to identify the vesicles. So in A, we are having a protein leaving the ribosome, entering into the rough endoplasmic reticulum for modification. In B, the vesicle from the rough endoplasmic reticulum is going to be moving to join the Golgi body. So in C, the vesicle or Golgi vesicle buds off from the Golgi body and moves through the cell. In D, the vesicle is being used in exocytosis. Remember, exocytosis, we are having releasing contents outside the cell. So the vesicle fused with the cell surface membrane and released whichever proteins it was carrying to the outside of the cell. Roman number 2, name one molecule or structure which leaves the nucleus by route E. We looked at examples of molecules that will be leaving the nucleus. Hope you remember some. We talked about the ribosome. The ribosome leaves the nucleus because remember it is made by the nucleolus, so it has to leave the nucleus to the outside. Okay, so we have the ribosome. Someone can talk about the mRNA. Roman number 2, through which cell structure must the molecule or structure you named in Roman number 2 pass through? The envelope, to get through the envelope. What is the answer to that one? Roman number 3, which structure should the molecule we named pass through? Yes, it should pass through the nuclear pore. So the answer is the nuclear pore. Name the molecule which leaves the mitochondrion in order to provide energy for this cell. Okay, we have not yet looked at the mitochondrion, but we know that a mitochondrion releases energy in the form of ATP. So in that case, for Roman number 4, the answer is ATP. The answer is ATP. That takes us to the mitochondrion. Usually it has such a diameter of 1 micrometer. It is surrounded by 2 membranes. We have this outer membrane and the inner membrane, which is folded to form cristae. You can see the folding of the inner membrane. In this case, this is a picture from an electron microscope. It still shows us the mitochondrion. You can see the outer membrane, and then these extensions are foldings of the inner membrane. So it has what we call cristae. Cristae are the foldings. Sometimes they are long, other times they could be shorter. And when you look at this mitochondrion, its major role is to carry out aerobic respiration, releasing energy for the cell. So it uses molecules such as glucose to be able to release energy in the form of ATP, which is adenosine triphosphate. But even if you just write ATP, yes, ATP is an energy carrying molecule. So once ATP has been made by the mitochondrion, ATP leaves the mitochondrion and goes to other parts of the cell where it can release that energy to be used for different chemical reactions. So the process of respiration can occur in the matrix. The matrix is like the cytoplasm of the mitochondrion. And it can also occur on these extensions of the inner membrane, the foldings of the inner membrane on the cristae. So that is why the inner membrane is folded to simply provide a large surface area for the process of respiration. Apart from carrying out aerobic respiration, the mitochondrion also synthesizes lipids. It synthesizes fats, it synthesizes oils. Yes. So it has 2 roles, aerobic respiration and synthesis of lipids. So usually cells that need a lot of mitochondria, like some people go to the gym and they make muscles, they lift weights. I don't know that we have some people around who go to the gym. So you find that those guys are going to have much more mitochondria in their muscles. Why? Because all the time they are needing energy to carry out whichever activities they do in the gym. And then we have the lazy ones who just sit at home to watch Netflix, So if you're the kind, you just sit at home, you have less mitochondria because you don't need as much energy as the other person who goes to the gym. Now, we need to look at the endosymbiotic theory. What is this theory about? When scientists observed mitochondria and chloroplasts, they found out that these two organelles had ribosomes that were smaller in size but and similar to the ribosomes in bacteria. When you look at the cells where the mitochondria and chloroplasts are found, the cells have bigger ribosomes, ATS ribosomes. But the chloroplasts and mitochondria have smaller ribosomes. So they were wondering, why is it that the ribosomes of mitochondria are similar to those of bacteria? So they further examined the mitochondria and chloroplast and found out that they do not only have ribosomes that are similar to bacteria, but even other structures within these mitochondria and chloroplast are similar to those found in bacteria. An example, they found that they also have small circular DNA molecules. And their DNA is exactly similar to that of bacteria. So that brought out an idea that, you know what? These mitochondria and chloroplasts are related to bacteria. So it was later proved that they were bacteria, ancient bacteria that were engulfed by the eukaryotic cells so that they can live together in a symbiotic relationship. Like the eukaryotic cells didn't have mitochondria before, but they engulfed them and the mitochondria became part of their body. For the plant cells, because they have chloroplasts, they engulfed chloroplasts and chloroplasts became part of them. So today, mitochondria and chloroplasts are part of those cells and they cannot live without each other because they are in a relationship, in a mutually beneficial relationship. Some of you are in relationships, and I could ask, is your relationship mutually beneficial? Is it symbiotic? Or it is parasitic? Anyway, so it was shown that mitochondria and chloroplasts were once bacteria, but now they are part of these eukaryotic cells. What shows? Number one, they have small ribosomes, 70S ribosomes. Secondly, they have DNA, circular DNA, and they can make their own proteins within a cell. They can make their own proteins. And at the same time, they can divide, they can replicate independent of the cell. They can replicate on their own. So that is it about the endosymbiotic theory. One other organelle we need to learn about are the microtubules. Yes, the microtubules. So microtubules are long, rigid, hollow structures. They are found in the cytoplasm. Yeah, this is a cytoplasm. This is a cell. Now, these yellow structures that are radiating outwards are what we call microtubules. They are really very tiny in size, 25 nanometers. So what are the uses of the microtubules? Micro means something small. So they are like very tiny, tiny tubules. And they make up the cytoskeleton. You know the way you have your own skeleton that has bones, you have a skull, even cells have their own skeleton. So their skeleton is made up of microtubules. So they form the cytoskeleton. Cyto means cell. Cytoskeleton, providing structural support to the cell, the way you have your own skeleton that provides you with support. And they have another use. They form an intracellular transport system. Intra means inside, intracellular, inside a cell. So inside a cell, we have seen an example, Golgi vesicles could move from the cell to the cell surface membrane to fuse so that they pour contents outwards. So if these Golgi vesicles are to move, most of the time they will be moving on top of these microtubules to move from one part of a cell to another. And that is why one of the uses of the microtubules is to form an intracellular transport system. Then membrane-bound organelles within a cell, those organelles will be held in place so that they stay in position by the microtubules. Microtubules also form spindle that is used in mitosis to separate chromatids during nuclear division. You'll learn about the details of that later. And at the same time, microtubules make up cilia and flagella. They make up cilia and flagella. We are going to look at cilia and flagella, and they also make up centrioles. They make up centrioles. Next, let's look at the microtubule structure. What is it made up of? So let's look at the structure of microtubules, the details of what microtubules are made up of. So microtubules are made up of a protein called tubulin, and this diagram shows you the tubulin proteins. These two proteins, alpha tubulin and beta tubulin. One is blue, one is dark blue. So it's called a dimer because it's made up of two proteins, two molecules, alpha tubulin and beta tubulin. So to make a microtubule, those dimers are assembled together. They are assembled to form what you call a protofilament. Now, we can use this to understand what a protofilament is. So this is one protofilament. You can see this protofilament is made up of just these tubulin molecules lined up end to end. This is another protofilament, another protofilament, another protofilament, and another protofilament. So those protofilaments are usually 13. 13 protofilaments are assembled together around a hollow core. And those 13 protofilaments form what we call a microtubule. Now, this means that a microtubule can be disintegrated very easily, and at the same time, it can be assembled very easily. To disintegrate, the way these dimers were joined to form a protofilament, they can simply be like disintegrated, broken down. So the assembly of those microtubules, either the assembly or the disintegration, happens in a region inside a cell known as a microtubule organizing center. A microtubule organizing center. Okay, from this we are noting that microtubules are made up of tubulin proteins. Microtubules can be assembled so that they are formed. At the same time, they can be disassembled or disintegrated within a cell. Like you'll learn that during cell division, spindle microtubules are formed. That means they are formed by joining these tubulin proteins into protofilaments. However, at the end of cell division, the spindle microtubules have to be disintegrated, broken down, because their role is already done. And there is a special region inside a cell that is responsible for assembling or disintegration of those microtubules, which they call a microtubule organizing center, MTOC. So we saw that one of the uses of microtubules is to form centrioles. So a centriole is another organelle that is found inside a cell. Centrioles are only found in animal cells. You never find them in a plant cell. They are only found in animal cells, and usually they are two centrioles. These centrioles lie close to each other, And that region where they are found is what they call a centrosome. So if this is our centriole, this region where it is found is what is called a centrosome. Microtubules form centrioles. Centrioles are made up of microtubules. So this is one microtubule, another microtubule, another one, another microtubule. The microtubules are arranged in triplets. So every centriole is made up of 9 triplets of microtubules. If we count them, we have one. This is our number 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets of microtubules. So each centriole is made up of those triplets, and they are surrounded around a hollow cylinder, a hollow tube. And they are around 500 nanometers in length and 200 nanometers in diameter. So each centriole contains 9 triplets of microtubules. Let's look at an electron micrograph that shows us a centriole. I don't know that you can see these triplets. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. So this is a centriole right here with the 9 triplets. This is in a transverse section, and then this is in a longitudinal section. What are the uses of these centrioles? These centrioles are used in production of cilia. So they form basal bodies where cilia originates from for organisms that have cilia on their bodies. And here they are showing us a picture of a cilium. So at the base of a cilium is a centriole, which is called a basal body. And here they are showing you the details of this centriole. You can see the 9 triplets of microtubules, meaning this is a centriole, but it's very special, found in cilia or flagella, and it's called a basal body because it's where the cilium originates from. These microtubules are said to be these centrioles are said to be microtubule organizing centers. Like an example, within this cilium, this basal body is responsible for forming these microtubules that form the cilium. So it organizes, it assembles the tubulin protein so that this cilium is made, because the cilium is also made up of microtubules. So centrioles act as microtubule organizing centers, especially in cilia and flagella. Now we have a centrosome as another organelle. Remember, a centrosome is that region where centrioles are found. So that centrosome also acts as a microtubule organizing center, especially during the formation of spindle, during mitosis, during nuclear division in an animal cell. So let's look at the cilia and flagella. Cilia and flagella are usually long, cilia are shorter. Cilia and flagella are made up of microtubules. These are whip-like structures that are going to be found in cells. Most of the time they're used for movement. Usually cells that have cilia, the cilia usually are many on a cell, but flagella, you could find some cells have just one flagellum, or they could even have more, maybe like 2. But the flagella are not as numerous as the cilia. So we are expected to be able to describe the structure of a cilium. Yeah, you are excluded from describing the structure of a flagellum, but at least you should be able to describe that of a cilium. So this is the structure of a cilium in transverse section. First things first, remember that a cilium is also made up of microtubules. But for a cilium, the microtubules are in pairs. You can see the pairs here. So this is a pair, another pair, another pair, another pair. So we have microtubule A and microtubule B, microtubule A and microtubule B, microtubule A and microtubule B. If you count the pairs, you're going to find that there are 9 pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 pairs. 9 pairs of microtubules. So those pairs are called doublets. So 9 doublets. Double means 2. 9 doublets of microtubules. Now, when you look at each doublet, each microtubule doublet is made up of A microtubule and B microtubule. A microtubule has all the 13 protofilaments. Remember, we saw that a microtubule is made up of 13 protofilaments. Like if you count these ones, if we start with this one here, number 1, number 2, number 3, number 4, number 5, number 6, number 7, number 8, number 9, number 10, number 11, number 12, number 13. You can see that microtubule A has all the 13 protofilaments. But when it comes to microtubule B, they are less. If we start with number 1 here, 2, 3, 4, 5, 6, 7, 8, 9, 10. Microtubule B has only 10 protofilaments. And microtubule A has arms. These arms are protein in nature, and they are made up of a protein called dynein. Dynein protein. So, microtubule A has arms, and these arms are very important during movement. For the cilium to beat, they make use of these arms. These arms on this microtubule have to touch this other microtubule. This arm will touch this one, this will touch this one, this will touch this one, and as a result, all the forces of the different arms touching and moving along the microtubule bring about the beating and movement in cilia. Next, in the center of the cilium, we have there microtubules that are single. They are called singlets. Singlets. So that is why you'll be told that a microtubule is made up of a 9 plus 2 arrangements. 9 plus 2 arrangement simply means it has 9 doublets of microtubules and then the 2 singlets, or the 2 microtubules that are found in the center. Now remember that a cilium is a cell structure. It is surrounded by a cell surface membrane. Remember, if this is an organ, sorry, if this is a cell, and maybe this is a cilium, it's an extension of the cell surface membrane. If that is a cilium, this is a cilium, it's an extension of the cell surface membrane, meaning those different parts of a cilium are found inside a cell surface membrane. That's why you see here we have a cell surface membrane. So I've talked about the features of a cilium. We have seen it has 2 central microtubules that are ringed in a ring, presenting a 9 plus 2 structure where we have 9 microtubule doublets outside and 2 central microtubules in the center. We have seen each microtubule doublet has, when you see this word MTD, it means microtubule doublet. It has an A and B microtubule. We saw that the wall of the A microtubule is a complete ring of 13 protofilaments, whereas that of B is a complete ring of, an incomplete ring of 10 protofilaments. We saw that each microtubule has an inner and outer arm. We saw that inner and outer arm, and inner, we are referring to the arm inside, and then outer to the arm outside. Those arms are made up of a protein called dynein. I think I didn't write the right spelling, but this is the spelling, dynein. So they connect with the B microtubules during beating. So that whole cylindrical structure, this is a cilium. So this whole cylindrical structure is called an axoneme. So this axoneme originates from a basal body. Remember, we mentioned that the basal body is made up of centrioles. So usually for cilia to be made of flagella, centrioles have to replicate. They divide so that they can form more cilia or so that they can form more flagella. Yes. So this is an axonym, this cylindrical structure with the 9+2 arrangement of microtubules. So these microtubules have a beating mechanism. They carry out motion. An example, you're going to find them along your gas exchange system in the trachea, in the bronchi. The cilia will be beating mucus that has trapped pathogens. Now, this is a scanning electron micrograph of cilia in the respiratory tract. That is maybe in the trachea or the bronchus. You can see the cilia originating from these cells. So usually for the cilia to beat, movement is caused by those arms. We saw that the arms make contact with the neighboring microtubules and they start moving along those neighboring microtubules. So that movement produces a force that makes cilia to beat and bend as they sweep. An example, as they sweep mucus away from your respiratory tract, or as they sweep, like another region where another region where cilia are found. Cilia are going to be found in the oviduct. You learned about reproduction in the oviduct where they beat the egg so that it goes in the region where it has to be fertilized. Even after a zygote has been made, the zygote has to be swept up to the uterus. And the cilia in the oviduct will be responsible for making sure that that zygote or the egg moves. So those are the uses of cilia. They are used for locomotion and in vertebrates like us, the beating cilia maintain flow of substances. An example, they ensure that mucus is removed from your respiratory tract to the back of your throat and then you swallow it. It's a protective mechanism for the gas exchange system. So that has been the outer structure of an animal cell. Let's look at the outer structure of a plant cell. Now, plant cells have features that are common and are also found in animal cells. So the roles features play in animal cells will be the same roles those features play in a plant cell, especially features that are common. We looked at the microtubules, we looked at the rough ER, we looked at the nucleus, we looked at the mitochondria, we looked at the smooth ER. So all those features play the same roles, the ribosomes, the Golgi body, the roles they play in an animal cell will be the same roles that they play in a plant cell. So in this case, we are just going to look at additional features which are not found in animal cells. So this is a natural structure of a plant cell as seen under an electron microscope. You can see the chloroplasts. You can see the ribosomes, the mitochondria, and we also have this is now an electron micrograph, a transmission electron micrograph showing us two plant cells. Actually, there are like three plant cells. We have this one, 1, 2, 1, 3, and they're showing you the different features in black and white. Remember, you should be able to relate what is here to what is this side so that you're able to identify the different features. You can see this is the nucleus. These are the chloroplasts. This is a cell wall. This is a vacuole. So let's look at those different organelles or features and their roles. We are starting with the chloroplasts. So chloroplasts are elongated in shape. You can see that they are really long and they are surrounded by two membranes. They are surrounded by two membranes. This is an outer membrane and an inner membrane. Still, if you go back to our previous slide, you can see the outer membrane and the inner membrane of that chloroplast. Yes, so chloroplasts have an envelope, an outer membrane and an inner membrane. So you just need to know what chloroplasts are used for. Chloroplasts are the sites for photosynthesis. They have the pigment, chlorophyll, or even other pigments, although the most common is chlorophyll, that is going to be responsible for trapping light energy so that glucose is made. Then the glucose can be turned into starch for storage. That is why within a chloroplast, you'll find there are starch grains. Starch grains are used for storage of starch. We also earlier saw that chloroplasts are similar to mitochondria in a way that they have 70S ribosomes and circular DNA. Remember that after sugars are made, some of them are turned into lipids during photosynthesis, so you still find lipid droplets. Lipid droplets in a chloroplast used for storage of lipids, which lipids can later be used in making membranes. Yeah, in A2, you will learn about the details of the structure of a chloroplast and what it is used for as far as photosynthesis is concerned. But for this level, you just need to know that it's used for photosynthesis. It has an envelope that is in an outer membrane. It has, so for it, its cytoplasm is called stroma. We saw that of a mitochondrion was matrix. And then it can have starch grains. It has circular DNA. It also has lipid droplets. Another organelle is the cell wall. You can see this structure here. Animal cells do not have cell walls, but plant cells have cell walls that are made up of cellulose. So we have this cell wall. It can be containing cellulose, but sometimes it can have other polysaccharides, maybe such as pectin, such as lignin, or hemicelluloses, but the most common is cellulose. So after the cell wall, you'll find the cell membrane. Now, in this case, the cell membrane is pressing against the cell wall, meaning this cell is turgid. So what is the use of a cell wall? A cell wall performs different roles. One of the uses is the fact that it's able to provide structural support to a plant cell, and it even determines the shape of a plant cell. How is it able to provide structural support? It has this cellulose. Cellulose is a strong molecule. You later learn about the characteristics of cellulose. It's a really very strong molecule that is able to provide strength to any structure where it is found. So it provides strength to the cell wall, and as a result, the cell wall can provide protection to the inner parts, and it can provide support and determine the shape of a plant cell. That's why plant cells have a definite shape, because of the cell wall containing cellulose. The cell wall has other roles. You can see that within this cell wall, where two neighboring cells meet, their cytoplasmic strands known as plasmodesma when one, plasmodesmata when many. So the plasmodesma allow exchange between neighboring cells. An example, materials can move into this cell and then also move from the other cell into this other cell. So plasmodesma are found within a cell wall. So when you ask for uses of a cell wall, you can say they contain plasmodesma that allow exchange between neighboring cells. One other role of a cell wall is that it provides a pathway for transport of molecules. What does that mean? If this plant cell absorbs water, assuming it has absorbed water and water has entered into the cell wall, some of that water can move through the cell wall from this cell and then enter into the cell wall of the next cell. And that movement of water through cell walls is called the apoplast. So apoplast is the movement of water within a plant, but via cell wall. So water can move from one cell to the next just through the walls. Yeah, just through the cell walls, which we call the apoplast. So cell walls allow water to move from one cell to the next through the apoplast. And maybe one thing I hadn't mentioned, if materials are moving from the cytoplasm of one cell via the plasmodesmata to the cytoplasm of another cell, we call that the symplast. So cell walls still allow the symplast pathway where materials move from one cell to the next through the plasmodesma, or they also allow the apoplast where water moves from one cell to the next just through the walls. Those are some of the uses of the cell wall. We have seen it provides mechanical strength and support to plant cells and a plant as a whole. Oh, I hadn't mentioned this. Cell walls prevent cells from bursting when water enters into them by osmosis, because they are really very strong. They prevent cells from bursting. You'll learn about osmosis later in chapter 3. Sorry, that will be chapter 4, not chapter 3. In chapter 4. And we have seen they determine the shape of a cell. We have seen that cell walls provide transport route for water and inorganic ions that could be dissolved in the water, which is the apoplast pathway. We have seen the plasmodesma in cell walls provide a transport pathway still for materials from one cell to the next, which we call the symplast. Now, these other uses, you'll learn about them in other chapters. So this one will be in chapter 7, where you learn about transport in plants. You learn about the endodermis. So the cells in the endodermis have a waterproof substance that prevents water from passing through the apoplast. However, you'll be able to relate when you reach that topic. Yes, you'll be able to relate when we reach that topic. And then you've learned about leaves. Leaves lose water through transpiration. But when you look at the epidermal cells of a leaf, they have a waxy proof layer of cutin, which forms the cuticle. And that cuticle is made on top of the cell walls of epidermal cells. So that cuticle prevents water loss by evaporation. At least I'm sure you know about the cuticle. So just pick any 3 that are easy for you to remember. You might not really cram all of them, but just pick any 3. You'll be good to go. Yes, you will be good to go. Yes, this lesson is really long, but if you're tired, you can pause and maybe start from there the next day or after taking a break. But my aim is to finish all the cell structures in plants and animal cells. So plant cells also have vacuoles, and in this case, we are looking at this large permanent vacuole. Animal cells do not have a vacuole. They do not have a large permanent vacuole. They have smaller vacuoles, which we saw the vesicles, yeah. But plant cells have this large permanent vacuole. And yes, we need to know the use of a vacuole. Usually when water enters into a plant cell, that water collects in this vacuole. And the vacuole keeps on bulging and bulging and bulging as more water enters into it. So it starts applying pressure on other cell contents. Before you know it, the cell contents are going to be pushed towards the cell wall. So it contributes to making plant cells turgid. Whenever it is inflated with water, whenever the vacuole has a lot of water, it builds up a pressure which makes the plant cells turgid. And turgid cells provide support to plant cells, especially to plant cells and to plants generally, especially plants that lack wood. That is why if plants do not have enough water, those that do not have wood, you find them wilting. Why? Because their vacuoles are not inflated with water. But whenever you provide them with water, you find them going back to standing upright because their vacuoles are inflated with water. What are the other uses of vacuoles? Plant cells do not have lysosomes, but their vacuoles act as lysosomes. They will be containing hydrolytic enzymes. We saw what lysosomes do. So some vacuoles act as lysosomes in plant cells. So plant cells can have one large vacuole or even more vacuoles. Yeah, so some of them can act as lysosomes. Vacuoles are used for storage of different substances. They can store different chemicals that plants make. You know that plants have the ability to make different chemicals. Like we have anthocyanins. Anthocyanins are pigments that are made within a plant. Like you find pigments in petals, petals having different colors. You'll find fruits having different colors. So those colors come from the pigments stored, and those pigments most of the time are in vacuoles. Vacuoles can even store different chemicals like tannins and alkaloids. Those are chemicals that prevent plants from being eaten by herbivores. You know how herbivores eat plants? So some plants wouldn't want to be eaten. That is why they'll have some chemicals in their vacuoles which herbivores detest. As a result, herbivores will not eat them. So these vacuoles store food. You'll find them storing sucrose. In seeds, you'll find them storing proteins. So they act as food reserves where food is stored. They store waste products. You will not find plants excreting that they have a kidney, that a plant wants to go and ease itself. No. So where do they store their waste products? So most of the time, their waste products are stored in those vacuoles. Yes, like here they're giving us a waste product known as calcium oxalate. It will be stored in a vacuole. These vacuoles allow a plant to grow in size. Whenever plants take up water, their vacuoles expand, and as a result, the volume of a plant cell also expands, allowing elongation and growth in size within a plant. So those are the uses of vacuoles. Now, this shows us a plant cell as seen under an electron microscope on the left, and a plant cell as seen under a light microscope. So please, you should still be able to tell the structures which can be seen in a plant cell with an electron microscope, but maybe not with a light microscope. And this is still similar to what we saw in an animal cell. We can still see the nucleus with two membranes. This side it has one membrane, nuclear pore, no nuclear pore this side, rough endoplasmic reticulum, non-existent this side, ribosomes, non-existent, mitochondria with two membranes, this side it's not seen. You can see the chloroplast with its double membrane, this side, you'll not be able to see that. You can see the chloroplast grana and thylakoids, this side you will not see it. So you should be able to tell and also draw some of these features in case you're asked to draw them. In case you're asked to draw them. Let's do this. Let's do this simple task. It's going to be our last section for today. It's a simple assignment. They have given us the rows of different organelles. All we have to do is to identify which organelle performs that role. Yeah, so we have A, B, C, D up to L. And let's test ourselves. Are we able to tell which organelles perform those roles? Okay, let's get started. Which organelle manufactures lysosomes? Which organelle manufactures lysosomes? Okay, that is going to be the Golgi body. Did you get it right? Next, we have which organelle manufactures ribosomes? Manufacturing of ribosomes. Which organelle? Nice, nucleolus. Is that the answer you had in mind? Okay. Next, which organelle synthesizes proteins? The ribosomes. Brilliant. Ribosomes. Okay, we go. Which organelle, which cell structure can bud off vesicles which form the Golgi body? Can bud off vesicles which form the Golgi body? Nice. The answer is rough endoplasmic reticulum. Someone could ask, can't I just write RER? Yes, you can, but I've seen sometimes in the mark scheme where they say ignore RER if a student has written RER and an examiner is told to ignore. So if possible, just write the whole word, rough endoplasmic reticulum. Okay, next. Which organelle can transport newly synthesized proteins around a cell? Newly synthesized proteins around a cell. We saw that proteins are transported in, yes, you're right, in vesicles. Yeah, in vesicles. Okay. Next, they are telling us which organelle manufactures ATP in animal and plant cells. Nice, that's mitochondria or mitochondrion in singular. Mitochondria. Controls the activities of a cell because it contains DNA. I'm sure everyone knows that. Which organelle controls cell activities? And that is the nucleus. Clap for yourself. Yes, it's the nucleus. The nucleus. Which organelle carries out photosynthesis? I would be heartbroken if you really can't answer this. Photosynthesis, yes, you're right. That is the chloroplast. That is the chloroplast. Which organelle can act as a starting point for the growth of spindle microtubules during cell division? So in that case, they're asking for the microtubule organizing center for spindle microtubules. We saw that is the centrosome. Centrosome, but sometimes I've seen where a student is still given centrioles because centrioles can also be microtubule organizing centers. So centrosome or centriole, but the most appropriate is centrosome. Contains chromatin. What is chromatin? Remember, chromatin is just loosely coiled DNA. And which structure contains DNA in that case? So it's still the nucleus. Partially permeable barrier, only about 7 nanometers thick. Partially permeable. Yes, that is the cell surface membrane. Cell membrane. Organelle that is about 25 nanometers in diameter. I told you take note of these sizes of the different organelles. Like we know the smallest organelle with just 25 nanometers, we saw it was the ribosome. Ribosome, not rico, ribosome. So guys, that is it about the actual structure of a plant and animal cell. Thank you for being around. Please subscribe to my channel. invite your friends to come and study with us. I'm here to take you through all the AS Biology. AS Biology for the Cambridge syllabus. Yes. So thank you for being around. See you again on the next one. Bye.